that cell with mutation has to survive. And then there will be clonal expansion. So there will be production of more cells carrying the same mutation, clonal expansion. And then there will be tumor progression. In tumor progression, on top of the original mutation, let's say it's mutation A, and there are 1,000 cells already, there will be formation of subclones. So on top of mutation A, 100 cells will have mutation X, another 100 cell will have mutation Y, another 100 cell mutation will have mutation Z, and so on and so forth. So there will be formation of subclones. Over time, the tumor will be more aggressive, and there will be accumulation of more mutation. So a solid tumor that is clinically apparent, napapalpate, does not have, contains the cells there, has many mutations. Hindi lang isa. Hindi lang isa. But there will be one mutation that is common to all of the cells in the tissue, and that is the original mutation. For a tumor to be clinically apparent, for solid tumor to be palpable, it has to be at least, it has to have a weight of at least 1 gram, and there are about 10 to the 9th cells per liter. At 10 to the 9th cell, 10, 10 raised to 9th power. So yeah, I can see the piece of the wait lang, lecture. Okay, so there are four classes of regulatory genes that are principal targets. The growth promoting proto-oncogenes, the tumor suppressor genes, genes that regulate apoptosis, and genes that are involved in DNA repair. So for proto-oncogenes, these are genes that encode for proteins that will promote the cell to proceed to undergo, to proceed with the cell cycle, hence cellular proliferation. And so for this gene to participate in carcinogenesis, there must be a gain of function mutation. A mutated proto-oncogene is known as oncogene, and that oncogene will encode for oncoproteins. Next is next are tumor suppressor genes. So these genes encode for proteins that apply brakes in the cell cycle. And so for these genes to participate in carcinogenesis, they must suffer from loss of function mutation. Loss of function of the gene, no tumor suppressor protein, no protein that will apply brake in the cell cycle, hence proliferation. Next are genes that regulate apoptosis. So depending on the role of the gene, the protein encoded by the gene, pwedeng gain of function or loss of function mutation. And then genes that are involved in DNA repair. For the cell to, to have the damage, dapat hindi siya ma-repair for it to participate in carcinogenesis, then there must be loss of function mutation in the genes that are involved in DNA repair. So kapag DNA repair na yung problem, walang repair, this will lead to acquisition of more mutations at an accelerated rate. So with this, the cell undergoes this mutator phenotype state, leading to genomic instability. So again, kanina dun sa first, proto-oncogenes, growth-promoting genes. Unmutated yon. The mutated counterpart are called oncogenes. oncogenes encoding for oncoproteins. So haploinsufficiency, so define lang natin. In haploinsufficiency, there is loss of only a single tumor suppressor gene allele, and this reduces the activity of the encoded protein enough to release the brake on the cell proliferation and survival. So what are the mutations that contribute to the development of malignant phenotype? We call them driver mutations. The first mutation, the first driver mutation that will initiate carcinogenesis, we call these mutations initiating mutation. Initiating mutation. So any mutation that contributed to the development of malignant phenotype is a driver mutation. The first of the driver mutations is the initiating mutation. Now, there are mutations na walang phenotypic consequences, walang effect sa phenotype ng cell. Walang effect. And so, kung walang effect sa phenotype, we call these mutations passenger mutation. So, walang effect sa phenotype, passenger mutation. Once established, tumors evolve genetically during their outgrowth and progression under the pressure of Darwinian selection. So survival of the fittest. Selection of the fittest cells can explain not only the natural history of cancer, but also changes in tumor behavior after therapy. In addition to genetic mutation, epigenetic aberration. So pag epigenetic kasi there is no change in the sequence of the DNA, right? So epigenetic alterations, aberrations also contribute to malignant properties of cancer cells. So what are these epigenetic modifications? DNA methylation. So in DNA methylation, once a DNA is methylated, that gene is silenced. So there is silenced gene expression. Another epigenetic modification is histone modification. This may either enhance or dampen gene expression. Now, there are 10 fundamental changes in cell physiology which are considered hallmarks of cancer. So we will discuss 8 dito sa discussion natin ngayon. Genetic instability and cancer-promoting inflammation are enabling characteristics that accelerate acquisition of genetic and epigenetic alterations that confer malignant hallmarks by promoting cell transformation and subsequent tumor progression. So ito yung i-discuss natin in details. Self-sufficiency in growth factors, insensitivity to growth inhibitory signals, altered cellular metabolism, evasion of apoptosis, limitless replicative potential, sustained angiogenesis, ability to invade and metastasize, and ability to evade the host's immune system. So again, ulitin natin, self-sufficiency in growth signals, nandyan pa rin yung proto-oncogene. So they promote cell proliferation, cell growth. So normal genes, unmutated proto-oncogenes, when they suffer genetic aberrations, they will be called oncogenes and they will encode for oncoproteins. Pro-growth proto-oncogenes may encode growth factor receptor, signal transducer, transcription factors, or cell cycle components. So kung makikita nyo, matitrace nyo dyan, ititrace nyo yung flow ng information from outside the cell. The information, the message is for the cell to grow, to proliferate. So lahat madadaanan yan. Growth factor, the receptor for the growth factor, signal transducer, transcription factors, cell cycle components. So these are the steps in cell proliferation, just a review. First is the binding of the growth factor to its specific receptor. Then with that, there will be transient and limited activation of growth factor receptor and signal transducing proteins. There will now be signal transduction to the nucleus. And with this, there will be induction and activation of nuclear regulatory factors, leading to DNA transcription and eventually expression of factors for entry and progression into the cell cycle. And later on, cell division. So in a normal process, growth factor, the cells need growth factors for them to proliferate. So this is the growth factor receptor. You have here the growth factor. They must bind to their respective receptor in order to stimulate the cell to proliferate. And this happens in a paracrine fashion. Paracrine. Pag autocrine kasi is, ako yung nag-produce, mag-a-act din sa sarili ko, yung molecule. Pag paracrine, I will release the molecule, it will act on the nearby cells. Pag endocrine, sa malayo na cell. So, in a normal cell proliferation, it's usually through paracrine action. Paracrine action. What happens in cancer is that the cells may acquire ability to synthesize the same growth factor to which they are responsive. Gumagawa siya ng sarili ng growth factor. And so with this, hindi na paracrine loop. Kasi I have the receptor, I will be making my own growth factor, and those growth factors will act on the receptors, on my receptors. So autocrine loop like in glioblastoma and astrocytoma, PDGF. They have PDGF receptor, they will make PDGF to act on the receptor. And in sarcomas, transforming growth factor alpha and EGFR. Parang doon na rin yung pinag-usapan natin kanina. So, kinase is transiently activated by binding of specific growth factor, followed by receptor dimerization and tyrosine autophosphorylation. Kasi yung kinase, ang ginagawa nila, ang role nila lagi is to phosphorylate. So there will be tyrosine autophosphorylation of several substrates that are part of the signaling cascade. Yun yung usual na nangyayari. In oncogenic or mutant receptors, there can be constitutive, meaning kahit hindi mo i-activate, kahit hindi mo stimulate, this will happen, constitutive dimerization and activation without binding to the growth factor. So yung receptor, they are constitutively activated and they will continuously deliver mitogenic signals to the cell. So these are examples of diseases where there is constitutive activation of growth factor receptors. In lung cancer, we have two here. The first is a point mutation involving ERBB1 gene that encodes for EGFR, epithelial growth factor receptor. That's a point mutation on ERB1, ML4, ALK. So gene rearrangement naman ito, nagkakaroon ng fusion ng EML4, ALK. This is seen in lung cancer. And then gene amplification in ERB2, which encodes for HER2 protein or HER2 new protein. This can be seen in patients with breast cancer. So this gene amplification of HER2 new, HER2, is commonly requested together with ER and PR, estrogen receptor and progesterone receptor. inaalam natin if uh, we have receptors for these hormones, estrogen and progesterone. Kasama niyan is this test to determine for gene amplification of ERB2. So here, so we're done with the growth factor receptor. So signal transduction naman. So signal transducing proteins. A good example is RAS, a mutated RAS. So even if you do not have the growth factor, even if receptors are not constitutively activated, if you have a mutated transducing protein, you have a mutated gene for transducing proteins, then continuous signals can be delivered to the cell for the cell to proliferate. So yung RAS gene, of which there are three, HRAS, KRAS, NRAS. So these RAS encode for RAS proteins. They are members of a family of membrane-associated G proteins that bind guanosine nucleotides, guanosine triphosphate and guanosine diphosphate, similar to the uh, larger trimolecular G proteins. They normally flip back and forth between an excited signal transmitting state in which they are bound to GTP. So active, they are bound to GTP. So nag-flip back yan to an inactive state. Mabilis lang yan, transient lang tong activation. Mabilis lang siya. So what will promote the uh, hydrolysis of GTP for it to return to an inactive form? That is accelerated by GTPase activating proteins or GAP here. So para bumalik siya sa inactive state. So ano yung mga possible na problem dito? If you have gain of function mutation of RAS, if you have loss of function mutation of GAP. Wait lang, charge ko lang go back. So this RAS mutation is the most common type of abnormality involving proto-oncogenes. So KRAS, yung KRAS,
may mga mutated in colon cancer, lung cancer, pancreatic cancer, HRAS in bladder cancer, urinary bladder, and kidney, and NRAS in melanomas and hematologic malignancies. So ano bang effect ng constitutively activated RAS? Continuous signaling from RAS. So downstream, it will affect several pathways. Sa MAP kinase pathway at saka PI3K pathway. So either way, ang effect ay cell growth, cell proliferation. Because if you activate MAP kinase, it will activate MYC or MIC. This is a transcription factor and cell cycle component decyclines. So leading to cell cycle progression, cell proliferation. With MYC, so just like in PI3K pathway, nagkakaroon ng pro-growth metabolism at increased protein synthesis. So promoting cell growth. So cell growth, cell proliferation. Now, there can also be alteration in non-receptor tyrosine kinase. Kasi we have receptors with intrinsic tyrosine kinase activity. Meron ding receptors na walang intrinsic na tyrosine kinase activity. That they will have to recruit another molecule to do the phosphorylation for the signal transduction to proceed. So, example nito, ito, in CML. So, in CML, ang sikat dito na pinag-uusapan is Philadelphia chromosome. Ano yung mga involved na genes? We have from chromosome 9, the ABL gene, and chromosome 22. Na nandun ng BCR gene. So, BCR. So, paano ba nangyayari dito? Yan. You have here chromosome 9 and chromosome 22. There will be translocation here, but the translocation will not involve both the BCR and ABL gene. Hindi. Kasi BCR will stay in chromosome 22. Magsistay siya sa chromosome 22. So, ang ABL ang lilipat. So, with this, the protein that will be produced, ABL kinase, will be constitutively activated dahil sa pagkaka-fuse niya with BCR. It's a kinase. And so, anong ginagawa ng kinase? Phosphorylate. It will continuously phosphorylate. So, increase kinase activity. Now, this causes CML, chronic myelogenous leukemia. So, in chronic myelogenous leukemia, the cells, if you are going to look at a patient with CML, if you're going to look at the blood, you will see that the white blood cells are in all stages of maturation, from the most immature blast to the most mature neutrophils. Lahat kompleto. Bawat stage merong representative. Pinakamadami ay sa myelocyte and mature neutrophils. Pero lahat makikita natin. Kasi ang pathology here is continuous cell proliferation. Hindi mapigil na cell proliferation. We said earlier that when malignancies are diagnosed, hindi lang isa yung genetic aberration, hindi lang isang mutation. There will be several in a mutation in the cell. Pero meron tinatawag na oncogenic addiction. This is in the treatment of patients with CML. Because what they have observed is, if you treat the patient with focus on one genetic aberration, the translocation here, confusion, BCR-ABL, if the treatment is focused on that, uh, fusion of genes. Even if there are other mutations, basta bigay mo lang yung medication, focus on one genetic aberration, there will be drastic improvement in the patient. Kahit hindi mo na-address yung ibang mutation. So, lumalabas na yung behavior, activity of the tumor cells are dependent on one oncoprotein only in this case. So, ang tawag natin sa phenomenon na yan is oncogenic addiction. So, other alteration in non-receptor tyrosine kinase. So, kanina translocation, but ito naman ngayon point mutation in individuals with polycythemia vera, primary polycythemia vera, essential thrombocytosis, and primary myelofibrosis. So, kung titingnan ninyo, in, sa final grading period, sa WBC na topic, you will realize na isa lang pala yung pathology dito in CML, polycythemia vera, essential thrombocytosis, and primary myelofibrosis. Under sila, in fact, in hematology, under sila sa isang group of disorders, myeloproliferative disorders. Kasi because of the genetic aberration, dire-diretso lang pala yung effect ng proliferation. Dire-diretso yung proliferation ng cells. So in polycythemia vera, it's panmyelosis, meaning WBC, RBC platelets increased. Pero pinakang malaking karamihan ng manifestation because of the increase in the red cell. Kaya mataas din ang hemoglobin ng patient. The same is true with essential thrombocytosis, mataas naman ng platelet. And this is secondary to mutation in JAK2. So we have receptors na walang tyrosine kinase activity, intrinsic tyrosine kinase activity, right? Kaya ang mangyayari, nagre-recruit tayo ng molecule. And dito, it is a molecule that's under the JAK-STAT pathway. So JAK2. This JAK2 with mutation can be constitutively activated. Kapag mutated yung gene, they can produce JAK2 that are constitutively activated, and they will continuously phosphorylate STAT. So what is STAT? Signal transduction and activation of transcription. So it's both a signal transducing protein and a transcription factor. So pwede siyang pwede siyang pumasok sa nucleus and transduce ng mitogenic signal. So again, that STAT can also act as transcription factor. Ano pa yung mga transcription factors that can be involved? The master transcription factor is MYB. The others are MYB, USE, FOS, and REL. So MYC proto-oncogene is expressed on almost all eukaryotic cells. It can activate expression of genes that are involved in cell growth, like cyclin D. They can upregulate expression of ribosomal RNA. They can upregulate expression of telomerase and reprogramming of somatic cells into pluripotent stem cells. So this is the cell cycle. So G1, S, G2, M. So meron tayong dalawang checkpoint. G1S at saka G2M. Here, kung pag-uusapan natin yung tumor suppressor proteins. So tumor suppressor proteins, P53 and RB. Ang P53, Saan siya involved? Sa G1S. And ang RB, G1S din. Pero ang P53 is also involved sa G2M, hindi lang sa G1S. Char. So for these molecules to participate in carcinogenesis, so yung cyclins, cyclin D, these are cell cycle components. Cell cycle components, so cyclins and cyclin-dependent kinases. Cyclins will have to bind with CDK, cyclin-dependent kinases. For these kinases to do their job, ano yung gagawin nila? Phosphorylation. Phosphorylation to promote cell proliferation. Now, meron ding, kung may nagpo-promote ng function, so these cyclin D dependent kinases, they're proto-oncogenes kung hindi sila mutated. And for them to participate in carcinogenesis, they must suffer from gain-of-function mutation. Now, there are molecules that apply brakes in the cell cycle. Nabanggit mo nga kanina, yung sa G1S, you have RB, P53, and sa G2M, P53. And for these molecules to participate in carcinogenesis, since they apply brakes, para mag-promote ng cancer, they must suffer from loss of function mutation. Okay. Dito na tayo. Balikan natin yung so RB gene. So this is a tumor suppressor gene encoding for tumor suppressor protein, RB. So RB protein is a negative regulator at G1S. At G1S, paano sila magpa-participate in carcinogenesis? If there is loss of function mutation in both RB genes, RB alleles, if there is a shift to inactive or hyperphosphorylated state by gain of function mutation or gene amplification of cyclin D, cyclin-dependent kinase. So, paano yan? So, this is yung RB. Ang RB rin yan. They are active if they are hypophosphorylated. Hypophosphorylated. So if they are active, they apply brake so there is no transcription. So kailan sila magiging inactive? If they are inactive, they will not apply brake. So there will be transcriptional activation. That will happen if they will be phosphorylated. Dito, they are phosphorylated. So RB is inactive if they are hyperphosphorylated. So sino nagpo-phosphorylate sa kanila? Cyclins, cyclin-dependent kinases upon binding with cyclin. So cyclin-dependent kinase 2 upon binding with cyclin E, cyclin-dependent kinases 4 and 6 upon binding with cyclin D. Cyclin D. So kung titingnan nyo dito sa illustration na to, so if they are hyperphosphorylated, no break, cell cycle progression, cell proliferation. If they are active, hypophosphorylated, they will apply brake, no transcription, so no cell proliferation. So kailan sila magiging inactive? So hyperphosphorylated form, if there is gain of function mutation of cyclin B, cyclin E, CDK4, 6, CDK2. Now, these cyclin-dependent cyclines and cyclin-dependent kinases, inhibited yan by CDK inhibitors P16 Inc4A. Now, this inactivates this. So if there is loss of function mutation of P16, if it's absent, then there will be increased activity of CDK cyclin, increased phosphorylation, inactive RB, hyperphosphorylated kasi siya, so inactive RB. So there is no block in transcriptional activation, cell proliferation, cell growth. So another way by which RB can participate in carcinogenesis, kung yung RB and its um, yung mga processes na may kinalaman sa RB is loss of cyclin-dependent kinase inhibitors, P16, now viral oncoprotein inhibition of RB, like in HPV E7, when it binds to RB for proteasomal degradation, and to cyclin inhibitors for inhibition. P53, TP53, this is the guardian of the genome. The gene is located on short arm of chromosome 17, region 1, band 3, subband 1. And this P53 is said to be the most common target for genetic alteration. The most common target for genetic alteration. They can stop neoplastic transformation by, first, transient cell cycle arrest. Transient cell cycle arrest. Second is initiation of apoptosis. And the third is hindi na transient, so permanent cell cycle arrest, and that is senescence. So yan ang ginagawa ng P53. It promotes cell cycle arrest, which is very important kasi it allows the cell to undergo repair kapag tumitigil siya. So transient cell cycle arrest, apoptosis, or permanent cell cycle arrest, or senescence. APC, adenomatous polyposis coli, is also a tumor suppressor gene encoding for APC protein, which is gatekeeper of colonic cancer. Because APC, this APC down-regulates catenin. This down-regulates catenin. In the absence of APC protein, it causes continuous WNT signaling and entry of beta-catenin to the nucleus, hence cellular proliferation. In the absence of this APC, so walang APC, beta-catenin gets inside. That will lead to cell proliferation. So other tumor suppressors, ecadherin in gastric cancer, particularly in the diffuse type, NF1 in neurofibroma, NF2 in schwannoma, VHL in renal cell cancer, as well as pheochromocytoma. P10 in patients with Cowden syndrome, WT1 in patients with Bilm syndrome, and PTCH in patients with Korlin syndrome and basal. Now, pangatlo, so tapos na tayo sa self-sufficiency in growth signals, sa insensitivity to tumor suppression. Third is altered cellular metabolism, Warburg effect. Tumor cells exhibit glucose hunger. They shift to aerobic glycolysis, So with high levels of glucose uptake, increased conversion of glucose to lactose. So mataas ang kanilang intake ng glucose. And this is a feature of malignant cells that is actually exploited by by technology yung PET scanning. PET scanning. So ginagamit nila is labeled glucose, fluorodeoxyglucose. Fluorodeoxy hanap nila by dividing cells. So not much with the energy, pero yung magagamit nilang mga macromolecules para palakihin yung cell or mag-proliferate ang cell. Yun ang priority nila. Now, in the state of dormancy, another source of energy for them, for these cancer cells, aside from yung napuprovide from the blood, ay yung autophagy. Yung sarili ng parts, yung ginagamit na to produce energy. Ontometabolism. So, bago mga discoveries lang ito, nakita nila na merong mutation in enzymes in Krebs cycle. Particularly yung isocitrate dehydrogenase point mutation. IDH point mutation. So here, there will be loss of IDH function. Nagpapalit yata. Saan ba yun? Okay. May kulang na slide. Sorry. Sige, balikan natin yun later. Yung part na yun. So in altered cellular metabolism um, in cancer cells, so there is a crosstalk between cellular metabolism and pro-growth signaling factors. So examples yan yung sa PI3 kinase AKT signaling. There is upregulated activity of glucose transporters and multiple glycolytic enzymes. In receptor tyrosine kinase, attenuation activity of M2 isoform. So M2 isoform of pyruvate kinase. This one converts phosphoenol pyruvate to pyruvate. And with MYC, with this mutation in MYC, there is upregulated multiple glycolytic enzymes and glutaminase. Kasi di ba sila nga ay involved sa pro-growth metabolism. Pro-growth metabolism. Babalikan yung illustration kanina sa RAS. So fourth is evasion of apoptosis. So evasion of apoptosis. The intrinsic pathway is the most frequently disabled. So intrinsic pathway is the most frequently disabled. Mas marami ring processes na involved in the intrinsic pathway na pwedeng gamitin to evade apoptosis so that the cell with its mutation will stay alive. So ano yung mga possible na um, processes that will be involved in the intrinsic pathway? We have loss of p53, reduced egress of cytochrome C, loss of AFAP, and unregulated inhibitors of apoptosis protein or unregulated IAP. Compared with extrinsic pathway, so dalawa lang yung kanyang processes na involved to evade apoptosis. First is reduced level of CD95 or FAS. And second is inactivation of death-induced signaling complex. So of all the processes, sa lahat ng mga processes nandyan, so intrinsic pathway is the most common sa dalawang pathway. Pero sa lahat ng processes na involved, may dalawang mechanism, may dalawang major na mechanism involved in cancer development that has something to do with evasion ng apoptosis. First is loss of PP53. Because if there will be loss of P53, there will be no upregulation of BAX and PUMA, which are pro-apoptotic. And second is overexpression of PCL2, anti-apoptotic gene. So, involved yan sa cancer survival. So next hallmark is limitless replicative potential, the evasion of senescence. Paano mo may evade na ang pagtanda? So, ang sabi, to promote senescence, there must be upregulation of tumor suppressors such as P53 and INC4A P16. Or you maintain RB in hypophosphorylated state. Kasi with that, mas nag-favor tayo with cell cycle arrest at G1S checkpoint. So to prevent senescence, kinakailangan i-disrupt mo itong mga processes na to. You have to disrupt these RB-dependent G1S cell cycle checkpoints. To have limitless replicative potential too, we have to evade mitotic crisis. We have to evade mitotic crisis. The problem kasi is, gusto natin mag-proliferate yung cell. But continuous proliferation may also lead to cell death because of mitotic crisis. Paano mapipigilan ng mitotic crisis? Kasi with continuous mitosis, you lose nucleotides sa terminal ends ng chromatin. You lose nucleotides. And to prevent that from happening, there has to be upregulation of telomerase or alternative lengthening of the telomeres. Of these two, the second is accomplished by DNA recombination. Of these two, the former, which is upregulation of telomerase, is the most common mechanism to evade mitotic crisis. The third, to have limitless replicative potential, is to have cancer stem cells. So for self-renewal. The sixth hallmark is sustained angiogenesis. So sustained angiogenesis. Tumors with the size of 1 to 2, at least 1 to 2 millimeter, will need their own blood supply. Otherwise, the cells in the tumor will undergo necrosis. So kailangan nila ng sariling blood supply. And for this to happen, there has to be angiogenic switch. So wherein there is a change in the phenotype of the cells that induces angiogenesis. And the processes here are mostly controlled by hypoxia. So paano magkakaroon ng angiogenic switch? And magkakaroon ng angiogenesis. First is the loss of inhibitors. What are these inhibitors? Angiostatin, endostatin, and vasculostatin. Second is increase in angiogenic growth factors. What are these growth factors? Vascular endothelial growth factor and fibroblast growth factor. VEGF and FGF. So hypoxia stabilizes HEAF alpha, which can activate transcription factors for VEGF and FGF. And decrease P53. Decrease P53, this will lead to angiogenesis kasi normally P53 stimulates expression of anti-angiogenic molecules and repress expression of VEGF that is pro-angiogenic. The seventh hallmark is the ability to invade and metastasize. So paano sila nag-invade and metastasize? Two phases. The first is invasion of the extracellular matrix. Ito yung original na tumor. They will invade the extracellular matrix. And the second is vascular dissemination. Papasok sila sa loob. And homing of the tumor cells. Mag-home, pupunta siya sa ibang area, lalabas siya. Mag-home siya dito, and colonization. So yan ang two phases. So invasion occurs in 4 steps: loosening of cell-to-cell contacts, degradation of extracellular matrix, attachment to novel extracellular matrix components, and migration of tumor cells. So we have metastasis gene. Ang tawag sa kanilang dalawa, snail and twist. These snail and twist, they promote the epithelial to mesenchymal transition. So epithelial to mesenchymal transition. Processes that are involved here are downregulation of cadherins. So downregulation of cadherins and upregulation of mesenchymal markers. So in invasion, for tumor to detach from one another, there has to be, nabanggit natin kanina, downregulation of cadherin, downregulation of E-cadherin. So naglulusen yung attachment nila with each other. In normal cells, this will promote anoikis. Anoikis is apoptosis due to loss of adhesion. But this does not happen in cancer cells. Hindi siya nangyayari. So aside from detachment of tumor cells from one another due to downregulation of cadherin, there will be degradation of extracellular matrix. Paano nangyayari to in cancer? Because of the secretion of proteolytic enzymes. Secrete yung matrix metalloproteinases, cathepsin B, and urokinase. Next is attachment to matrix components by making use of molecules, integrins attaching to fibronectin and laminin. And next is locomotion. Paano mag-move ang cancer cells? Due to cytokines from tumor cells, stromal cell motility factors, and matrix components. So growth of metastatic deposits. Growth of metastatic deposits. Ano yung mga factors that can affect the growth of metastatic deposits: location and vascular drainage of the primary tumor, uh, tropism of particular kind of tumor cells for specific tissues. There are tissues that are really not, based on observations, hindi sila fertile soil for um, colonization of cancer cells like skeletal muscle. Hindi nyo commonly maririnig na nag-metastasize saan sa skeletal muscle. So walang ganun. And then escape from dormancy, like yung PTHRP, parathyroid hormone-related protein from breast cancer cells to stimulate osteoblasts to make rank L to activate osteoclast to have osteoclastic activity. So for organ tropism, so nabanggit na natin yon, yung skeletal muscles, hindi sila favorable na site for um, colonization ng cancer cells. Ano pa yung pwede mag-affect ng organ tropism? Adhesion molecules on endothelial cells of target cells. Adhesion molecules and chemokine receptors like CXCR4 and CCR7 on breast cancer cells. Next hallmark is the ability to evade the host immune system. Tumor cells can be recognized by immune system as non-self and can be destroyed. They can be mediated by cell mediated mechanisms, although tinitingnan nila na yung antibodies, kung ang antibodies ay pwede, pero wala masyado on antibodies silang nakikita sa studies. So more on cells. So yung antibodies provide, based on recent studies, little evidence of providing protection. So ang lagi natin pinag-uusapan dito is cells, particularly lymphocytes, mononuclear cells, in immune surveillance. They guard against tumor development. The principal cell is the cytotoxic T lymphocyte. First line of defense is our natural killer cells. They can destroy tumor cells without prior sensitization. Macrophages can also participate, and they do so by producing reactive oxygen species. How do cancer cells evade the immune system? By selective outgrowth of antigen-negative variants for them to be undetectable ng mga cells na may receptor for the original antigen of the tumor cells. Next is loss or reduced expression of MHC molecule. Because if you do not have the receptor for, if you do not express MHC, kung wala kang MHC, then the cell will not be able to display antigen to cytotoxic T cell. Without the display, then the cytotoxic T cell will not be able to kill the cell that has been transformed into a malignant cell. Next is activation of immunoregulatory pathways to inhibit tumor immunity, like upregulation of CTLA4. As we know, the CTLA4 competes with CD28 on the surface of T cell. They compete with CD28 with CD28 in binding with B7 on the surface of the antigen presenting cell. And we know that if it will be CTLA4 that will bind to B7, this will lead to anergy. And so the cell will no longer be responsive. The T cell will no longer be responsive to the antigen that has been presented. Eventually, every time na makakasalubong niya ang cell, cancer cell, with carrying the antigen, that cancer cell will go unnoticed. Hindi siya mapapansin. Another mechanism to induce energy is PDL1, PDL2 binding on PDR on T cells. So ganun din siya. Ganun ang effect, energy. The fourth to evade the host immune system is secretion of immunosuppressive factors, transforming growth factor beta. So this is a molecule that is secreted when cells are um, when uh, secreted during repair phase kapag merong inflammation, pag magre-repair na. The fifth is induction of immunosuppressive regulatory T cells. Ano ba yung mga possible na antigens naman natin? No, ano yung mga possible na antigens? Remember that cells undergo mutation, non-lethal genetic damage. And with the mutation, there will be change in the sequence of the DNA. And so there will be protein variants. Protein variants na may express due to the mutation. These can serve as antigen, the protein variants. Second is yung overexpressed or aberrantly expressed self-protein. So overexpressed like, no, this is a normal molecule pero sobrang dami niyang express. Nagiging parang antigen na rin siya. Tyrosinase. So yung mga yan can serve as antigen. And in cells that are infected with viruses, oncogenic viruses, so there are proteins that can be uh, used inside the cell. Like if the cell is infected with HPV, the cell can have E6 and E7 proteins, EBNA in cells that are infected with Epstein-Barr virus. So these can serve as antigen too. Now, inflammation is said to be cancer enabling. So inflammation is said to be cancer enabling. Paano nasabi na inflammation is cancer enabling at nagpo-promote siya ng cancer development? Because remember, in inflammation, there will be tissue damage. And so with the tissue damage, there will be repair. There will be repair. And for these processes to occur, there has to be secretion of growth factors. There has to be secretion of growth factors. These growth factors can also act on cancer cells. They can also promote proliferation of cancer cells. Second is the removal of growth suppressors. Removal of growth suppressors. Cell to cell and cell to extracellular matrix interaction suppresses growth. Proteases can destroy the interaction by destroying adhesion molecules. Next is enhanced resistance to cell death. So resistance to cell death. Kanina nabanggit ko that for cancer cells to invade, they must first detach with one another. Down regulation of cadherin. Nabanggit ko rin kanina yung anoikis na in normal cells, anoikis should happen. They should undergo apoptosis once you lose the attachment to your neighbors. But in cancer cells, it does not happen. Why? Kasi tumor-associated macrophages prevent anoikis by producing adhesion molecules. So they produce adhesion molecules. Next is by inducing angiogenesis. Remember that in repair, you produce extracellular matrix, you also produce blood vessels. So there is release of pro-angiogenic factors in inflammation that can enable growth of cancer. Next is activating invasion and metastasis. So the molecules that are needed for cancer cells to easily invade, infiltrate the surrounding tissue can also be found in inflammation. TGF-beta promotes epithelial mesenchymal transition. Tumor necrosis factor and EGF promotes motility. Proteases will allow the cell to easily undergo locomotion in the matrix. Next is evading immune destruction. How will they evade immune destruction? Um, TGF beta, which is uh, upregulated in inflammatory processes, can promote Treg recruitment and suppression of cytotoxic T lymphocytes. M2 macrophages, they can promote angiogenesis, fibroblast proliferation, and collagen deposition. Hmm, Nandiyan pa ba kayo?
Yes po,
Yes,
Doc.
yes po.
Okay pa tayo?
Yes po, Doc.
Kasi there are 4 more maliliit na topic ito. Genomic instability, ilang page lang to. Genomic instability, dysregulation of cancer-associated genes, molecular basis of multi-step carcinogenesis, and chemical carcinogenesis. Yan. Iiwanan ko sa inyo yan as reading assignment. Hello?
Yes
Okay
po,
po,
Doc.
Doc.
Bigay ko sa inyo yung 1 hour for you to go through those pages. So remind ko lang kayo, na-approve yung request ninyo na magkaroon ng plating on Monday night, 6 to 7 p.m. So together with the plating on Neoplasia 1, lab quiz on Neoplasia 1, you will also have your SGD quiz 5. Tama ba? SGD quiz 5 on Monday night. Then next week, may upload ako na para siyang practical exam, pero it's a dummy exam na multiple attempts. Parang practice exam lang siya. It's a dummy exam, hindi siya recorded. So parang pwede nyo gamitin yun para mag-practice ng practical. So that will be next week. Ayan. So you have 1 hour and 6 minutes. You can go through the 4 topics. Genomic instability, dysregulation of cancer-associated genes, molecular basis of multi-step carcinogenesis, and chemical carcinogenesis. Okay?
Okay po, Doc.
Pwede kayong magstay sa Moodle, pwede rin hindi. Pwede na kayong mag-leave if you want. If you need a